today we're going to talk about count and tabular data. And count data are very common, as we all know, in sequencing studies. But that's not the only place where we have counts. For example, many of us, after we've done chip experiments or differential expression experiments, then go and try to do gene enrichment studies where we look at, say, GO categories or KEGG pathways and see if our significant genes are showing up as being enriched in certain of those categories. So in this talk, I'm going to talk about tables as a way of showing associations between count data and some kind of treatment or category or classification, and discuss some of the common effects found in tables, and talk about the two most common tests, which are basically tests of independence between the counts and the categorical variable, the chi-square test and Fisher's exact test. So, actually, what I'm going to do for a minute, I realize that all of my slides are like typical tabular data, but they're not the ones that we see every day. So I pulled up one of my papers, but I couldn't get the slides, I couldn't get the tables in very easily, so I'll just show you the tables. So this is typically what you get from an RNA-seq type study. L1 are the number of reads from the first gene, and L2 is from the second. And then this is an unreplicated study, so there's just a treatment and a control. So all the other ones are in the control, and then there's this thing called the margin, which is the total number of reads for the first gene, second gene, and so on. And here we have the library size at the bottom. Okay? And what we do with those data, and this will be fun to do in the paper, for each gene, we convert it into, can't quite see the whole table. You convert it into a table like that, so there's the ith gene, and there's the reads from that gene, and then you just sort of accumulate everything else into an other gene category, and you get a table with 2 rows and 2 columns. And then the question you want to ask is, basically, are there more or fewer genes than you would expect for this feature, say, in the treatment, given everything else in the table, right? And that's what we would test, and Fisher's exact test is typically done for that. And then the SNP table, SNP data usually comes as a 2x3 table, so at each SNP you have genotyped your sample, and you know whether it comes from the treatment and control. And now instead of reads, these counts here are the number of samples that come from this, are in this category. Okay? So typically, these, let's see, both the row and column counts here are much smaller because, you know, we have millions of reads and at most thousands of patients, right? But anyway, those are the kinds of tables we're going to be looking at, and this is called the 2x3 table because the edges, which are called the margins, don't count as the table. The table's the stuff in the middle. Okay. So I'm going to start with this table, which actually is just a table of samples. So this is actually one of the classic microarray studies of 2 kinds of leukemia. I think it's acute lymphomic leukemia. You guys know more medical stuff than me. And acute myelomic leukemia. Anyway, they did microarray studies to see what the genetic signatures were for these cancers, and here's where the patients came from. Okay, so hopefully you can see a problem in this table, especially based on what we said about experimental design last time. So what's the problem? yeah, basically, whatever DFCI is, and I can't remember which hospitals, what? Dana-Farber? Almost all the ALL samples came from there, and none of the AML samples came from there. So if there's any sample handling or patient population or anything else differences that could affect gene expression, and of course everything affects gene expression, it's confounded with the cancer type. And almost all the information on ALL comes from there. I mean, it's not even as if there's another hospital that has a lot of patients that came in both groups. This is basically it for ALL. And it has no AML patients. I mean, it probably has AML patients, it just didn't have any AML samples for this study. So immediately we have a problem. Whether this was a design problem or it just happened during the course of the study that these were the patients that came through, I don't know. But we're going to have some problems. And these kinds of problems should show up immediately once you tabulate data. This kind of confounding can lead to something called Simpson's paradox. So I pulled this right off Wikipedia for the example of Simpson's paradox. But what you notice is this. Overall, treatment B did better in terms of success rates, right? But in each category, treatment A did better. Treatment B did better overall because more of the readily cured patients got treatment B, not because treatment B is better for those readily cured patients. Everybody get that? Well, see, the small stones are more readily cured. You can see that in both treatments. And the treatment A got about a quarter of its patients in that category. The treatment B had 80% of the patients were in that category. So treatment B looks better, not because it's better for these small stone patients, but because more of the patients that got treatment B had small stones. That is, they weren't as sick, so they did better. And that makes the treatment look better. Yeah, that's called Simpson's paradox, and it actually happens with non-count data too. In non-count data, we call it a latent or lurking variable or covariable, something that you didn't measure that's affecting the outcome and is biasing the study. This is why we randomize, right? And in tabular data, if you start, obviously we can never use every single factor that we have in the study for analysis. If we did, we'd end up with these big, gender by treatment, by age, by large or small stone, by blah, 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 and we'd have one person in each category and we'd never be able to do an analysis. So Simpson's paradox basically says that lurking variables can affect your conclusions, and this is more prevalent in tabular data, or at least it's easier to detect in tabular data. Okay. So here's another example from the same study of another thing that happens with tabular data. So this is the patients tabulated by gender. Okay. And things look pretty equal, right? But there's something missing in this table, and it's actually the same thing that actually led to people allowing the Challenger to take off when the weather was really cold, leading to O-ring failure, which led to the first shuttle space shuttle disaster. But you guys are all too young to remember all that fuss. What's missing? The gender was not always reported. And we don't know whether there's something lurking there. Maybe they didn't report just for males, right? In which case, it's, if that's true, then maybe some of these cancers are more prevalent in males. So, okay, so make sure when you're doing things that you don't just take out data because the information isn't there. If the information isn't there, you need a separate category saying the information isn't there. One of the things in R, when R creates tables for you, there's actually an option you have to set to make sure that the missing data, which is called NA, not available, shows up in the table. So the key was, I didn't change the row sums here, but the key was that the total number in the previous table was bigger than 49. And that's kind of what they missed in this Challenger thing too. They left out all the flights in which there were no failures, which were all at high temperatures. And so it looked like there was no temperature effect. But when you added the information at which there were no failures, all of a sudden you saw that temperature had a strong effect because at low temperatures there were lots of failures. Well, not a lot, few, just not fatal ones until the Challenger. Often we do row and column percentages. So there's the row percentages. Each count is a percent of the rows. Column percentage, each count is a percent of the column. Overall percentages, each count over the total there. Now, the fundamental statistic for a 2x2 table is called the odds ratio. And often we take the log of the odds ratio. So if, how can I put this? PI is either the row or column percentage, and basically if the rows are independent of the columns, so basically that says the row percentages are the same or the column percentages are the same, then this statistic is 1 and its log is 0. Okay? So if they're not proportional, so that say males disproportionately get ALL, then this will be far from 1. Okay? And this is the same statistics that are used in case control studies where we look at risk ratios. You know, if you smoke, what's your risk ratio for lung cancer compared to a non-smoker, that kind of stuff. It's this statistic. And this little thing here just shows that it doesn't matter whether you use rows or columns. So in the top one, I use the column percentages, and in the bottom one, I use the row percentages, but I get the same answer. Okay, and what we're going to test when we test independence is whether this so-called odds ratio is 1. When we don't have a 2x2 table, we actually have 2 independent odds ratios, and so we have what are called 2 degrees of freedom for the test, and basically it's combining 2 odds ratios into a test. Yes?
Can you explain the odds ratio?
Okay, so the odds of something is a way of looking at the probability of it happening versus it's not happening. It's the same thing as odds in betting. Okay, so if the probability, so suppose you're an ALL in the ALL group, then the relative risk by gender is the same if you're equally likely to be male or female, right? And in that case, your P1 over 1 minus P1 will be 1. And if it's also the same for the AML patients, then P2 over 1 minus P2 will be 1. But that's not what this is saying. This isn't saying within gender are you equally likely, within males are you equally likely to have this cancer. It says, is the percentage of males that have this type of cancer versus the other one the same as the percentage of females that have this type versus the other one? And that's why, So basically it says, is the odds ratio in both groups the same? So that's that. So if the odds ratio is the same, then the statistic is 1. And if there's elevation in one of these, obviously if one of them is up, one of them has to be down, right? So if one of them is up, then then one of those 4 numbers will be bigger than expected. And another one in a diagonally opposite, either in the same row or the same column will have to be down relative to the others. And that will force the odds ratio either to be bigger than 1 or less than 1, depending on which one you have on top there. But just like with the t-statistic, there's variability. You don't, even if patients with ALL are equally likely to be male and female, as patients come in, you might at some point have 7 males and 9 females, right? So you need to do a statistical test to account for that variability. Okay. So the chi-square test is the one that we usually teach in Stat 100, and it's good when the counts are big. These counts may not be quite big enough for that. So the way that works is we say, well, suppose that odds ratio was 1. What would you expect these counts to be? Okay? And what you expect is that, 65% of all of the patients have ALL. So if there's no gender effect, 65% of the females should have ALL, and 65% of the males should have ALL, right? So you just multiply 65% by the number of females and 65% by the number of males, and that gives you the expected number of males and females that have ALL, and the same thing with AML. And so that's what you get. And if you use the column percentages instead, you get the same numbers. That's key to this. And now the question is, are there any deviations of these counts from what you'd expect? Okay, and what's the largest deviation here? Yeah, the AML with gender not reported. Right? But it's also one of the parts of the table that has the biggest counts. So you expect larger deviations where there's more stuff going on, right? So the question is it significant? And the way we're going to measure that is we take the differences. Okay, those are plus or minus, right? There's lots of ways we can force them to be positive, but the one that works statistically is usually squaring it. So we take the difference and square it. That makes it positive. And then you have to ask, what's the standard deviation? And it turns out that for count data, the standard deviation is usually, for this kind of table, is proportional to the expected count. The variance is proportional to the expected count. So the standard deviation is the square root of that. So now we have to square the expected count, so we want to square the standard deviation. I've said that all in words, and hopefully I've got the formula there. So there's the formula. So on the numerator are those squared deviations. And on the denominator is an estimate of the standard deviation of the squared, I haven't quite said that right. It's actually the square of like a t-statistic on the top. Sorry. It's the deviation over its standard deviation, all squared, is what I'm trying to say. Okay, and then we sum it over the entire table. And when all the expected counts are bigger than 5, and if the gender is independent of disease, then this thing has an own distribution called the chi-square distribution, and you can look it up on a table just like you can look up the t distribution, or you can get the computer to compute it for you. And in this case, The chi-squared statistic is 23, which turns out to be really out in the tail, so it's got a very small p-value. One test, right? We're only doing one. When we do multiple, you gotta still have to do multiple comparisons. And so we're going to conclude that there is some deviation in this table that's way out of line. Now, which one is it? Well, the statistic itself is made up of these little components, right? So the biggest of these components is the one that's making this thing big. Maybe there's two big components, but certainly the biggest one is going to be making this big. And it turns out that in this table, not surprisingly, it's the AML with gender not known. But that's not very good for us as biologists or medical practitioners, because it doesn't tell us anything except that the folks in, let's see, that AML was Dana-Farber, right? They didn't report gender. That's all it's really telling us. So what we probably want to do is get rid of that column. Now that we know that there's a problem, you know, we go back to the hospital and say, hey, you guys have to give us this information in the future. Right? And now we can do some biology, crossing our fingers that their data looks like everybody else's. Now we see that the chi-squared is very small for the rest of the table. The expected values are really, really very close to what you see. And the p-value is large, so this is a totally expected table. The only problem is now we have very small expected counts, and this chi-square test, the test statistic is fine. The distribution we're using to look it up is not fine, okay? It's based on a normal approximation. Anyway. So when we have this situation with small counts, so thinking about sequencing data, we always have some tables with small, we always have some genes with small counts, or some features with small counts. Basically, this chi-square test, it works great when we have large samples, and then when they're getting smaller, there's, you know, some mathematics you can do to make it a little more accurate, so it's still pretty good, but once you're down to expected values of like 4, 3, 2, it really doesn't work. So the next thing we do is to get an exact p-value using something called Fisher's exact test. In the old days, people didn't do it because it was too computationally intensive. But, you know, so even 25 years ago, it was pretty computationally intensive. But now, you know, you can do it on your iPhone easily. So the way that works is it takes the table and says, what are the tables that have exactly the same row and column totals, but that are more extreme in terms of same proportions as this table? Okay? So what I always like to do is focus on the smallest cell in the table. So what's more extreme? Well, the smallest cell there is 3. And there's 3 females. The simplest thing to focus on is gender, right? Because that has the smallest there. Okay, so what would be more extreme than having, what would be less extreme than having 3 of the 8 females have it, 3 of the 8 AML patients be female? What would be less extreme? 4 and 4. That's the only thing that's less extreme, right? Absolutely equal numbers. Everything else is more extreme. So that would be 2 and 6, 1 and 7, 0 and 8, and also 5 and 3, 6, right? So what we do is we compute the probability of all those tables. Okay? But every time, if I change the 3 to a 2, not only do I have to bump that up to 6, but I also have to bump the other one up to 21, because this has to stay fixed too. Okay? So that's why it was computationally intensive, because you had to figure out all these tables and figure out the probability that each table occurs. It turns out there's a nice probability rule. If you don't know what this is 23 choose R, which is the number of ways of taking 23 objects and choosing R of them, like 3 or 2 or whatever. So, so this thing is called the hypergeometric distribution, and sometimes this is called the hypergeometric or hyper-G test. In R, it's called the hyper-G test. And there's also a routine that's called Fisher's test, and it uses the hyper-G test to do the computation. So it computes it for all the values of R that are more extreme under the hypothesis of equally likely to be female, and comes up with a p-value. And here's what you get. So there's Fisher's exact test for this particular table. It got an exact p-value of 0.7. The chi-square test, which is approximate, got a p-value of 0.84. Okay. So it's off by a fair amount. And then this test also works directly on the odds ratio, so it gives you a confidence interval for the odds ratio. So before, remember I said if the t-test, if there's no difference between the means, the confidence interval will have 0 in it. So here, if things are proportional, the odds ratio is 1, so the confidence interval should have 1 in it, and it does. But it's also a very wide interval. It says that odds ratio could be as big as 11. Okay. And that's because there are not a whole lot of AML patients for whom we have the gender. We have a small sample size. It's not that we have a small sample size for AML, but because gender wasn't recorded, we ended up with a small sample size. Yes?
So would it be the same if you were then checking to see what would be less probable for ALL?
Well, it turns out that the table, the whole table adjusts together, so it would, you would come up with exactly the same test statistic, but the computation would be nastier because now instead of just having 8 things that I have to distribute into the 2 cells, I'd have whatever the total there was to distribute between the 2 cells, so I'd be summing over a whole lot of more things. That's why I picked the smaller one.
So you just select the smallest?
Yeah, because you should get exactly the same answer when you do the exact computation. Of course, on the computer, doing the same computation 2 different ways may not give you exactly the same answer due to round-off error, approximations, and formulas. But it should be extremely close, unlike these 2 things. But what you would find is if that smallest expected count had been 6 or 7, these 2 numbers would have been out only in like the 2nd or 3rd decimal place. Okay. So as I mentioned, we use this all over the place. We use it for gene enrichment analysis, and that's where you're going to see it first in this course. I think Carmen will be talking about that. He'll probably go over it with the hypergeometric test and that test. You see it in RNA-seq differential expression analysis and SNP frequency analysis, and so on. But there are some differences, some of which have to do with multiple testing, and some of which have to do with the nature of the data. So in gene set enrichment analysis, this might not be so much a factor when you're doing pathway analysis, because as far as I know, people don't look at nested parts of the pathway, but maybe that's wrong, maybe they do these days. But in gene ontology terms, there's a nested tree of gene ontology terms, and the way the analysis works is it kind of goes up the tree to find the highest node at which things are enriched. And that means not only do you have multiple testing, but you have multiple testing in this sort of funny nested thing. But most of the software packages available now do the multiple testing adjustment for you, so when you get a p-value from that, it's been adjusted. In RNA-seq, differential expression, hopefully you have biological reps, otherwise you're really only making inference about the 2 samples you're looking at. you don't really get a simple two-way table, so you have to account for the biological variation as well as the count variation. So we're going to use a more complicated model for that rather than just these tables. But at some points in the analysis, sometimes this is useful. So the data that I'm going to give you was an early study. It has 2 lanes per sample to get the read counts high enough. So the first thing I did was check whether the 2 lanes were the same, basically, proportional. And I used Fisher's exact test. Somebody yesterday said, well, they were worried about, they had multiplexed and they worried about the number of counts they had, so they took the controls and ran another lane of controls. And so there might have been a batch effect, and same thing. All you really need to do is take the 2 lanes of controls and see how they, whether they match up okay. That is, whether they're proportional to the library size, using Fisher's exact test. Here we have a big problem, the correlation among the p-values problem. The SNPs that are close together or in the same gene or the same promoter and so on are often correlated, and so these tables are correlated, and that's a much harder problem to deal with. But each individual test will still be a chi-squared or Fisher's exact test. It's a multiple testing issue, not a test for the individual table. And that's it. We might actually finish early today. I don't know, are there questions first, please? Yes?
We will be seeing some examples through the course of how this is applied to RNA-seq data.
So the first thing, I think the first place you'll see it is gene enrichment data from microarray studies, which Harmon will talk about after we talk about microarray analysis. And then you'll see it again when I go through this case study for RNA-seq data. And then I'm not quite sure what the CHIP guys will do, but it is used in CHIP also. But we're not going to do a lab today, because at this point it's just the 2 tests, and it's just kind of yet another set of R commands. Are there other questions? So do you want to have the break now, or do you just want to go right in?
Yes, what do you want to do?
It's only 9:30, so what do you want to do?